right? So we'll be discussing the hemoglobin part and the porphyrin. Now, guys, this is the exam series that has already been initiated from no July, June 1st, right? So you already know in this particular sessions, no, we are revising all the important questions, which are no very like potent questions for the upcoming exam. So we are revising all the important concepts in a in a very crisp and concise manner and conceptual manner too, right? So obviously this is beneficial for all the first year students who are going to appear for the university exam in August, right? Apart from that, the students who are appearing for the NEET PG exam and the FMG exam, they can also like listen to the lectures because I'll be covering all the important previous year topics along with the previous year questions, right? So yes, so these are the long questions and short questions that I will be covering up today. Now, you guys already know the core features of the Oracle app, right? So the live classes which I'm taking right now, it will be there on the Oracle app. Apart from that, if you miss the live classes, guys, you can go for the recorded classes. And guys, most importantly, the notes over here, the notes that I'm using in my live classes, they are all curated in a form of no Q&A based on question and answer format. Apart from that, after each and every topic, guys, you will get the MCQs, right? From basic to advanced level. So yeah, the clinical questions will be there, which are now being more relevant in the exams, right? All the previous no 10 years questions are being covered, like all the important PYQs, PYTs, everything is there. So yes, so that is the basic plan that you can see on the screen, guys, right? Apart from that, if you want some additional discount, you can use my code Dr. Abhijeet 20 for the further discounts, right? So let us just start with the very first question, that what is hemoglobin, guys, right? And what is the type, structure, and function? Normally, guys, this is a a long question that is being asked in the university exam. So let us just start with the basic definition of the hemoglobin. Now, something which you know from class +1 or +2, guys, right? That hemoglobin, it is the red pigment, guys. It is the red pigment, and it is exclusively found in the erythrocytes. Now, when I say erythrocytes, guys, you already know I'm talking about the RBCs, right? The red blood corpus cells. Okay, the red blood corpus cells. Now, guys, very importantly is the range of the hemoglobin in the blood, right? So you should know that, yeah, the normal level in males, right? In males, it is 14 to 16 gram per deciliter. And in females, guys, it can vary from 13 to 15 gram per deciliter. Obviously, if the values are lesser than 14 in males, or if the values are lesser in 13 in males, guys, that is something which is referred to as anemia. That is something which is referred to as anemia, right? So that is something which you have to like take away from here. Not this is not the anemia wala. If the values are lesser than 14 in males, and if the values are lesser than 13 in females, right, we have mild, moderate, and severe anemia, right? So that is something that you have to remember. Apart from that, the hemoglobin levels also varies no in pregnant females, right? In pregnant females, automatically the levels of hemoglobin are decreased. So that is the physiological change in the pregnancy. That is something not pathological, guys. So in pregnancy, guys, obviously the levels of these hemoglobin, they will be certain slightly decreased than the normal, approximately 11 to 12 gram. It is the normal like the range in the pregnant woman, right? So that is something which is called as the physiological change in the pregnancy, and that is completely absolutely normal in the these pregnant females. So that is some simply the physiological change in the pregnancy, right? So that is some key points that you have to remember over here. The normal hemoglobin levels in male and females. Apart from that, I have also talked about the pregnant females, and hemoglobin is something that is a red pigment that is exclusively found in the red blood corpus cells. Now, guys, now when we say, guys, when we say hemoglobin is a conjugated protein. Now, whenever I say conjugated, guys, conjugated means it is made up of not only protein, there has to be some other component also, right? So we have a protein part, that is the globin. Globin we have discussed yesterday, right? We have discussed the globin yesterday, that is the protein part, and heme, that is a non-protein conjugate part, and that is a non-protein conjugate part, right? So we have the heme plus globin. We have the heme plus globin. We have the heme plus globin. Now, globin actually it is a protein, right? It is a protein. Okay. Apart from protein, we have a non-protein part that is called as the heme. Now, what is heme made up of? What is heme made up of? So you already know the heme. It is a non-protein part. It is a non-protein part. And what it is exactly made up of, guys? So just write down if you have to like simply write down what is the heme made up of? So heme it is made up of a metal and porphyrin and porphyrin, porphyrin, right? So a heme is basically made up of a metal and a porphyrin. What is porphyrin? I will be discussing it in a couple of minutes. But metal, guys, just remember we have a metal over here that is called as ferrous, Fe plus 2, Fe plus 2. So hemoglobin is comprised of two words, right? The heme and the globin part. The heme, basically, it is a metal and a porphyrin ring, right? I'll be coming to the porphyrin part, but which metal is there in the hemoglobin? It is Fe plus 2. And which protein is there? That is the globin part, right? So heme, it is containing protoporphyrin 9 molecule, right? With one ferrous atom at the center, right? With one ferrous atom at the center. Okay, so that is porphyrin or protoporphyrin 9. That is combinedly called as the
Hello, sir.
um is it like continued, sir?
Sir, I will continue recording, sir.
Like, is it
Just
fine
fine.
with the internet, or shall I change the
No,
connection?
no, it's fine, sir.
Is it fine, right?
Yes.
Okay. Okay. So methynyl bridges. So methynyl bridges, guys, you already know it is double bond CH. Okay, that is the methynyl bridge, right? That is the methynyl bridge. So yes, if we talk about the no pyrrole ring, guys, if we talk about the pyrrole ring, so let us just try to draw the pyrrole ring, guys. It's like very simple, right? So yes, now okay. So yes, over here, guys, you already know whenever we say porphyrin. So porphyrin is basically we are saying that there are four pyrrole rings, four pyrrole rings. Basically, they are joined by methynyl bridges. Methynyl bridges, right? So porphyrin, these are four pyrrole rings joined by methynyl bridges, and methynyl bridges I have just told you it is double bond CH dash, right? Now, when we say pyrrole rings, guys, pyrrole ring, it is basically a five-membered ring. It is a five-membered ring, guys. It is a five-membered ring. So remember, guys, yes, this ring is just like this, yeah, containing nitrogen over here. So you can see the number 1, 2, 3, 4, and 5. This is a five-membered ring. And how many are the pyrrole rings? We have the four pyrrole rings. So we have to just draw it in the same manner, guys, over here, right? So yes, okay. So that is how you have to remember over here. We have to draw these 4 pyrrole rings, right? So these are the 4 pyrrole rings, and they are just joined together by these methynyl bridges, right? So remember, yeah, these are just joined together by these methynyl bridges, right? So that is something you have to remember. So that is how the structure looks like. That is how the structure looks like. That is something. And in the center we have an iron, guys. That is the ferrous ion. So these are the pyrrole rings. Pyrrole rings are 5-membered rings. Just I have drawn over here, and these are connected together by these methynyl bridges, and that is C double bond. And yes, that is something which you can see over here, which I have highlighted, right? That is how they're connected together. And in the center of this structure, you can find this iron, guys. In the center of the structure, we have the iron. We have the iron. So that is something which you can see over here also. Now, again, guys, remember that there are 3 types of porphyrins are there. I will be coming to this topic separately. So as of now, just remember there are 3 types of porphyrins are there, right? Based on side chains, based on side chains, right? Sorry, yeah, they will be like, yeah, 4 types based on side chains, right? Based on side chains. And remember, they are MPVA. Now, when I say M, it is methyl, propionyl, vinyl, and acetyl rise, right? So we have 4 types of porphyrins based on the side chains, and these are MPVA, that is methynyl, yeah, the methyl group, the propionyl group, right? The V, that stands for vinyl, and A stands for acetyl. So based on the based on the side chains, actually, we have these 4 types. So that is about the um, heme part, right? That is about the heme part. When I say hemoglobin, heme consists of iron, and these 4 pyrrole rings, which are linked together by these methynyl bridges. Apart from that, we have the globin. Globin, guys, you already know it is a protein chain. Globin is it is a protein chain. So you already know this globin actually basically belongs to a class of globulins. It belongs to a class of globulins, guys, right? It belongs to a class of globulins. And remember that hemoglobin, it is a tetramer, guys. It is a tetramer which contains 4 subunits, okay, which contains 4 subunit, and that is held together by some non-covalent bonds, okay. They are held together by the non-covalent bonds. And remember that each subunit, guys, has a globin chain and a heme group, right? Each subunit, okay, how many subunits are there? 4. And out of, like, each subunit will be having one, yeah, globin chain and a heme group, right? So that is about the main structure of the hemoglobin, right? Now, coming on to the types of the hemoglobin. Types of the hemoglobin. The very first one, guys, that is displayed on the screen, that is hemoglobin A1, HbA1, right? Now, HbA1, the first important question for exam, it is the major form of hemoglobin in adults. Again, a 1 mark Viva question, which hemoglobin it is the major form of hemoglobin in adults? It is HbA1, that is HbA1, hemoglobin A1. Now, again, second Viva question that can be asked in the practical exam, that how many amino acids basically make this HbA1? That is 574 amino acids and has 4 subunits. 2 alpha, that is 141 amino acids, and 2, yeah, that is 146 amino acids, right? Now, each alpha subunit processes primary, secondary, tertiary structures, right? So I have already told you we have these 4 subunits, and these 4 subunits are associated by these non-covalent salt bridges to form the HbA1. Okay, so these primary, secondary structures basically they try to form a quaternary structure, guys, and that is something resulting in the formation of this HbA1, right? Coming on to the next one, that is hemoglobin 2, so it has 2 alpha subunits and 2 beta subunits, right? Coming on to the third one, that is the fetal hemoglobin, guys. Very, very important. Fetal hemoglobin. Okay, now when I say fetus, guys, you already know I'm talking about when the conceptus is from 9 weeks to 40 weeks of pregnancy. That is something called as this fetus part, right? And this fetus, it is not having that adult hemoglobin. It is having some hemoglobin which is termed as HbF. Okay, that is the fetal hemoglobin. So it has two alpha subunits and two gamma subunits. And remember, if I compare the adult hemoglobin with the fetal hemoglobin, adult hemoglobin basically the okay, over here, the lifespan, it is basically 60 to 90 days. That is again very important, right? It is having 60 to 90 days. So you can very well see, guys, in the fetal hemoglobin, we have these two gamma subunits and two alpha subunits. Okay, and in the adult hemoglobin, we have the two alpha subunits and two beta subunits. So that is exactly the difference in the tetrad over here. Fetal will have two alpha units and two gamma units, but as we compare the fetal hemoglobin with the adult hemoglobin, the adult will have two alpha subunits and two beta subunits, right? Coming on to the functions of the hemoglobin, again, this is a fact-based question, guys. You already know from class +2 that it helps in the exchange of gases, right? Hemoglobin. The main function is to transport which gas, that is the oxygen, guys, right? And carbon dioxide, right? Now, you already know it carries the oxygen from the lungs to the tissues, all over the tissues, and from all the tissues, it collects the carbon dioxide, right? And it transports the blood, right, back to the lungs. So that is something you have to remember, the very primary important function, and that is something like everyone knows that the main function of the hemoglobin, it is to transport the oxygen, right, and the carbon dioxide for transport of oxygen from the lungs to different parts of the body, vice versa. From the tissues, it collects the CO2 and just get it to the lungs. Apart from that, you should remember it has a buffering capacity, guys. Buffering capacity, and buffering capacity is just basically due to the histidine content. That is something that you have to remember. So this hemoglobin also acts as a buffer, guys, and that is mainly coming from this histidine content. Apart from that, that is the second function of the hemoglobin that you have to remember. It is cooperativity, right? So remember now, if we talk about cooperativity, guys, hemoglobin exhibits positive cooperativity. Okay, why? Now, when I say cooperativity, guys, now what happens? See that if hemoglobin has like four subunits, right? Two alpha, two beta, right? If we talk about the adult hemoglobin, you guys already know if there is affinity of oxygen, if only one unit. Now we have two alpha, two beta, guys, right? Two alpha, two beta, and if one subunit of the hemoglobin, okay, if it is binded to the oxygen, it will increase the affinity of the other subunits also. Okay, it increase the affinity of the other subunits for oxygen. Now, this is something, this phenomena basically allows the hemoglobin to efficiently load and unload the oxygen. Now, there are four subunits in the hemoglobin, right? Two alpha and two beta. If one subunit, okay, is binded to the oxygen, now that what it will do, it will convince the other subunits to take oxygen. And if all the subunits will have oxygen, what is this called as? That is something called as the loading of the oxygen, right? So it allows the hemoglobin to efficiently load and unload oxygen as needed, right? So it allows the hemoglobin to efficiently load and unload the oxygen. Coming on to the next part, that is the oxygen saturation curve. I think this also is something which we dealt in +1, right? The NCRT book of biology, in the respiratory system, right? We already discussed that the hemoglobin's cooperative interaction with subunits results in a sigmoidal. Okay, well, what is the nature of this graph, guys? Now we are plotting this graph between the oxygen partial pressure on the x-axis. We are taking this oxygen partial pressure, and on the y-axis, guys, we are taking the oxygen saturation of the hemoglobin. We are taking the oxygen saturation of the hemoglobin. So remember, guys, the resulting graph, it is basically a sigmoid shaped graph. It is basically a sigmoid shaped graph, right? So now what is this graph is actually, so it is between the oxygen concentration, guys, right, and the hemoglobin saturation, okay, and the hemoglobin saturation. Now, this curve basically reflects the ability to adapt the affinity for oxygen, right? So with changing oxygen levels, right? So it can go to right shift and it can go to left shift. Now, whenever the no, this graph is shifted to the right shift, so there is decreased affinity for oxygen. Okay, everyone knows the very possible cause in higher altitudes, guys, obviously. So this graph, no, if I just want to plot a graph for the person who is on higher altitudes, obviously this graph will be shifted towards the right, and that is something called as the right shift, and that is due to the decreased affinity for oxygen. Obviously, oxygen will increase, the partial pressure of CO2 will automatically increase. CO2 increases, the acidic component will increase. Acidic component increases, the pH decreases, guys, right? So that is something that you have to remember. On the other hand, that is the left shift, that is the increased affinity for oxygen, right? So if uh okay, if there is increased affinity for oxygen, automatically the partial pressures of the CO2 will decrease, right? There will be decrease in the H plus, right? So yes, obviously, if there is decrease in H plus and decreased acidic component, now, you already know it will be shifting more towards the neutral or the alkaline component, right? So the pH, right? So that is something you have to remember over here, right? Coming on to the next question, guys, that what is meant by the allosteric regulation, guys, right? Now, remember, guys, the hemoglobin basically it results in no two forms, right? There are two transitional forms of the hemoglobin, the T and the R form based on the oxygen binding. The T form has the lower oxygen affinity, very, very important. Remember, guys, that the T form has a lower oxygen affinity, while the R form has a higher affinity, while the R form has a higher affinity. And this transition facilitates oxygen loading and unloading in different environments. Now, what are the factors that no affect? What are the factors that affect the hemoglobin function? So now, the factors which affect the favoring from T to R transition, high oxygen affinity. What is something which if you're going to high oxygen affinity, obviously the partial pressure of the oxygen will be increased. And if the partial pressure of oxygen is increased, automatically the partial pressure of carbon dioxide will be decreased, right? Apart from that, the factors which favor from R to T transition, that is the low oxygen affinity, obviously the partial pressure of this oxygen will be decreased and there will be increased partial pressure of carbon dioxide, right? And guys, one more important, that there is decreased pH, right? Decreased pH, right? That is the Bohr's effect. Bohr's effect, guys, right? Now, coming on to the next question, that is the abnormal hemoglobin. And the two things that I have to discuss, very, very important, that is the thalassemia and the sickle cell anemia. The thalassemia and the sickle cell anemia. Now, abnormal hemoglobins are something, guys, that is defined as the hemoglobins that are produced due to mutation in the genes. You have already read in class plus two also. There are mutations in the genes that no code for alpha and beta globin chains of hemoglobin. So we have alpha chain and we have beta chain. We have alpha chain and we have beta chain of hemoglobins, right? We have alpha chain and beta chain of hemoglobins. If there is a mutation in the alpha chain or the beta chain, right? If there is a mutation in the alpha chain and the beta chain, what will happen, guys? Automatically, the resulting hemoglobin will be not normal, it will be abnormal hemoglobin. And that abnormal hemoglobin is formed due to the mutation, and that mutation is basically in the genes that code for these alpha chain or beta chains, right? So remember, guys, the abnormal hemoglobins can be of two types. The very first one that you have to remember over here, that is due to the altered primary structure, or there could be. See, we have discussed the primary, secondary, and the tertiary. They know combine to form the quaternary structure, or it can be due to altered quaternary structure. Okay, because all these subunits are actually making up these, and that is something resulting in the quaternary structures, and that quaternary structure, if it is disturbed, it will lead to a disorder, something called as the thalassemia, right? So now, Let us discuss the first type, that is due to altered primary structure. From P, guys, just try to remember that this type is generally due to the point mutation, primary point mutation, right? Which results in the single replacement of a single amino acid, right? Leading to formation of hemoglobin with altered conformation. Obviously, guys, अगर एक point पर if there is no a change only at a point, what will happen, guys? Automatically the other sequence will be disturbed, right? And that is something which will result in the formation of an altered conformation. Okay, and that is why, guys, you have to remember some of the examples. The very first one is sickle cell anemia. Okay, the sickle cell anemia. So yeah, you guys know the glutamic acid at sixth position of beta chain, it is replaced by valine. Very, very, very important for the Viva question. I told you it's a point mutation, guys. It will be only changed at a single point, right? And automatically at which position, guys? At sixth position of beta chain. Now this is let us suppose this is the beta chain, and at sixth position, we have okay, we have sixth position, we have glutamic acid, we have glutamic acid. Now, if there is any mutation, guys, what will happen to this glutamic acid? It will be replaced by valine. It will be replaced by valine, and this results in the formation of a sticky patch on the surface of the beta chain, right? Of both oxy and deoxy hemoglobins, right? Now, what happens, guys, if no see if there is valine which is no getting attached to the sixth position, it is replacing the glutamic acid. What will happen? There will be something called as the sticky patch which will be no formed, right? So it will result in the polymerization of this hemoglobin, okay, forming the long fibers, and that is something which will distort the erythrocytal structure, or it will break down the erythrocytes, forming the sickle shaped RBC. Sickle shaped, okay, the sickle shaped RBC, as you can just see in the yeah, in the image also, right? The sickle shaped RBC. So that is something you have to remember. And remember, the sickle cell RBCs are very, very fragile. They can break very easily. Now, if, guys, if the RBC is broken, okay, if RBC breaks, what will happen? The hemoglobin, that is also altered, it will come out, and that is something called as hemolysis, the breakdown of the hemoglobin. That is breakdown of the hemoglobin. Obviously, in RBCs we have the hemoglobin. Obviously, if the structure of the RBCs is no getting changed, it is getting converted into these sickle shaped. And remember, these sickle shaped RBCs, they are more fragile, guys. They will break easily. And what will happen after the membranal membrane of the RBCs are break? What will happen? These hemoglobin automatically will come out, and that is something of no use. That is something called as hemolysis. Hemolysis means the breakdown of the hemoglobin. Okay, and that is resulting in anemia. Obviously, hemoglobin is not there. Hemoglobin is decreasing. So obviously, these patients of sickle cell hemoglobin, obviously they have anemia. That is something which is referred to as the sickle cell anemia. Anemia means low levels of hemoglobin. Obviously, the hemoglobin over here is lysed, or it is breaking down. So that is something you have to remember. Coming on to the next one, guys, that is hemoglobin C. Now, hemoglobin C, guys, you should know that the glutamic acid at sixth position, it is replaced by the lysine. That is again 1 mark important Viva question. Apart from that, we have the hemoglobin M. So a 58 histidine is replaced by tyrosine, right? So remember, the histidine, it is replaced by the tyrosine and hemoglobin M. And hemoglobin T, the glutamic acid at 124 first position, it is replaced by the glutamine, right? The glutamic acid at 121st position, it is basically replaced by the glutamine, guys, right? So that is about the different types of the hemoglobin. Apart from that, guys, let us just focus on one of the disorders. That is the thalassemia, guys. That is the thalassemia. And you already know these are abnormal hemoglobins that are basically altered due to the different altered subunits, right? So again, thalassemia, it is basically from one, or yeah, it is from one or more alpha or beta chains of hemoglobin due to a defect in the production of alpha or beta globin chain. So there are 4 copies of alpha globin gene, okay, 2 on each chromosome, 16, and 2 copies of globin gene, 1 on each chromosome, that is 11. 11, right? So we have chromosome 16, chromosome 11. Now, what is the different types of thalassemia, guys? The very first one, that is you already know. We have the alpha thalassemia, right? We have the alpha thalassemia. Now, alpha thalassemia is something which is caused due to a defect in the production of the alpha chain. Okay, it is caused due to a defect in the alpha chain, and we have like various no types of this alpha thalassemia. We have the carrier of the alpha thalassemia, we have the trait of the alpha thalassemia, we have the minor of the alpha thalassemia, and we have alpha thalassemia major, right? Now, alpha thalassemia major, it is basically hydrops fetalis, right? Absence of all four alpha globin genes. That is something resulting in hydrops fetalis, right? That is the absence of all four alpha globin genes, right? Apart from that, guys, remember if we say a carrier, guys, carrier is something which is only having absence of one globin gene, guys, right? There are no clinical effects in the carrier, guys, right? The carrier is something which will be passing on the genes to the further successive generation. But over here, remember, the carrier is something which will no, the carrier will have no presenting complaints, no clinical effects, but it will be having absence of one globin chain, right? The trait will be having absence of two globin chains, and that is something called as the benign condition, guys. That is the benign condition. Apart from that, minor is having absence of three chains, and major something which is having absence of all the four chains, right? So you have to just remember like silent, trait, minor, and major. Silent is something which will be having no only one gene which is absent, and there will be no clinical features. The trait having two globin genes, two alpha globin genes absent. The minor being the three globin genes will be absent, and the fourth one, that is the major one, all the four globin genes will be absent. Beta thalassemia, it is basically obviously due to the production of the beta chain. We have beta thalassemia major and minor. So remember, when we say major, it is both the beta globin genes are absent. And when we say minor, it is the one beta globin chain which is absent. So remember the minor one, it is a heterozygous condition, guys. It is a heterozygous condition. Whereas the major one, it is a homozygous condition because both the beta globin chains are absent in the major, whereas the minor one only one is absent. So that's why it is like different traits, right? So that is about this. Coming on to the next one, that is what are the hemoglobin derivatives, guys? So remember that the hemoglobin derivatives basically they result from the combination of various substances with hemoglobin. Now, guys, the major derivatives, the very first one, what is bounded to the hemoglobin? That is the oxygen. So the very first one that you have to remember, the derivative of the hemoglobin can be oxyhemoglobin, oxygen bound hemoglobin, right? Or we can have the deoxygenated blood, okay? That is deoxygenated hemoglobin, okay? While the minor derivatives basically they include carb amino hemoglobin, right? Apart from oxygen and deoxygenated part, right? The CO2 wala part. Basically, what else can bind to the hemoglobin? We have the carb amino hemoglobin, we have a methemoglobin, and we have a carboxy hemoglobin. And that is something which will result in disorders, guys. Normally people know these hemoglobin has affinity for oxygen and the oxygenated, but there are certain other some chances which can also bind to this hemoglobin, and that is the very first one that we have to remember, that is the carb amino hemoglobin. And remember, it is formed by the okay carb amino. When I say carb amino, I'm talking about the carbon dioxide, guys. I'm talking about the carbon dioxide combined with hemoglobin. Okay. Now, guys, remember it is responsible for transporting approximately, guys, it is around 2 to 10% of the total carbon dioxide in the blood. That is carb amino hemoglobin. So the carbon dioxide transportation in our body, okay, via the help of hemoglobin, it is only about 2 to 10%. It is only about 2 to 10%. Coming on to the next one, that is the methemoglobin. Methemoglobin, basically the hemoglobin containing iron is in the Fe+3 state. Normally, without the structure of the hemoglobin, in the center we have the ferrous ion. That ferrous ion it is basically in the form of Fe+2. But over here in methemoglobin, what is the altered structure from the normal? In the normal we have Fe+2. Over here we will have Fe+3. That is something called as methemoglobin, the met hemoglobin, right? Now what happens? This Fe+2, it is getting oxidized, guys. It is getting oxidized to Fe+3 via hydrogen peroxide free radicals, right? Via hydrogen peroxide free radicals, and that is See, in a normal person, guys, what is actually there? We have something called as the methemoglobin reductase enzyme. Methemoglobin reductase enzyme, which converts this Fe3 again to Fe plus 2, again to Fe plus 2, right? Because remember, guys, Fe plus 3 cannot bind or transport oxygen. It is only Fe plus 2 state. The structure of the hemoglobin in the center, we have Fe plus 2, and that Fe plus 2 is only, no, the only state in which we cannot transport oxygen. If it is converted to Fe plus 3, it cannot transport oxygen, cannot bind with the oxygen, as oxygen transport by hemoglobin requires iron in the ferrous state, only in the Fe plus 2 state. If it is converted into Fe plus 3 by some hydrogen peroxide radicals, again there is an enzyme in the body that is something called as the methemoglobin reductase, which can convert it back to Fe plus 2 and the process of transportation can carry on. So remember, what happens? What happens that there is a condition called as methemoglobinemia. Now, when I say methemoglobinemia, so methemoglobin, you already know it is methemoglobin, right? Emia means blood. So what is methemoglobinemia? It is increase in the methemoglobin levels in the blood. It is increase in the methemoglobin levels in the blood. So now, okay. Now, this can be caused due to certain drug side effects, right? For example, you take sulfonamides or exposure to certain compounds like nitrates, guys, very, very important. Or there is something called as aniline also, right? Or there is hereditary hemoglobin M. Okay, if from it's passed from generation, so that is the hereditary hemoglobin M. Or there is one more enzyme in the body that is methemoglobin reductase. अगर methemoglobin reductase काम ही नहीं करेगा, if it is not working normally, guys, what will happen if there is reduced activity of methemoglobin reductase? Will it be able to convert Fe plus 3 back to Fe plus 2? No, obviously. And Fe plus 3 cannot bind to the oxygen. It cannot bind to the oxygen. And that is something, guys, which will not no transport the oxygen to the blood. And that is, yeah. Now, how does a person appears of no methemoglobinemia? Whenever a person no comes, there is something called as cyanosis, guys. There is something called as cyanosis, right? Or I would say specifically, if we talk about methemoglobinemia, we have chocolate cyanosis. Chocolate cyanosis means all the no peripheral part of the body will be no brownish bluish in color, right? There will be brownish blue skin of the patient, right? That is something a very characteristic feature of the patients who are suffering from this methemoglobinemia. And that is apart from that, these patients, obviously the blood supply to the brain will be reduced. Patient will be coming to you with complaints of headache. The oxygen carrying capacity, the oxygen is not going to body parts. The patient will come to you with complaints of dyspnea. And obviously, they can be coma patient. If no oxygen, obviously the patient will land up in coma. And in severe cases, beta, the patient will die. That is something which will lead to death. So that is something you have to remember, right? So now, what is the treatment, guys? Which enzyme is defective over here? Yeah, methemoglobin reductase, because methemoglobin is the one which will convert the ferric to ferrous form, right? Fe plus 3 to Fe plus 2, it will again convert Fe plus 3 to Fe plus 2. So what we have to give these patients, we will give injections of these methylene blue to reduce Fe3 plus to Fe2 plus. And when it is converted to Fe2 plus, again it will bind to all the oxygen and the oxygen supply becomes normal, right? So that is something you have to remember. Apart from that, coming on to carboxyhemoglobin. Okay, now when I say carboxyhemoglobin, now hemoglobin derivative can also bound to carbon monoxide. Now guys, hemoglobin can also bind to oxygen and hemoglobin can also bind to carbon monoxide, which has higher affinity, guys. Again, a very important viva question. Carbon monoxide has a much higher affinity than oxygen, much higher affinity than oxygen. So whenever carbon dioxide, carbon monoxide levels are increased, no, in situations when there are burns or there are what will happen? The carbon monoxide concentration will be much more, right? So what will be there? There will be impairing of the formation of oxyhemoglobin because this carbon monoxide has higher affinity. And if it is like more in the surroundings, it will go and it will just bind to the hemoglobin, pushing the oxygen aside, just go away. So that is something that you have to remember. The normal blood level of carboxyhemoglobin is 0.16%, but it increases in smokers, the persons who are smoking, the continuous smokers, chronic smoking. What will happen, guys? This percentage of carboxyhemoglobin 0.16% will land up in 4 to 5%, guys, and that is something very, very severe and fatal, right? So that is something you have to remember. Yeah, obviously. uh, okay, let's imagine a patient, who is coming to me. He's saying that he's having history of 10 years of smoking. Daily he's smoking a packet of cigarette. And that is something, remember guys, this carboxyhemoglobin levels will automatically increase. That will be up to 4 5%. This patient will come to me with complaints of breathlessness. Second, nausea, vomiting, headache. Obviously, the oxygen carrying capacity will be reduced. The blood supply to the all the parts will be reduced, resulting in more symptoms like headache, drowsiness, chest pain will be there. Okay, breathlessness will be there, and if it is like continued, and if the patient is not listening to the doctor to quit smoking, ideally the first step is to quit smoking, the precautionary step. But precautionary step is never to start smoking. But what is something when the doctor when he comes to the doctor, apart from everything, I will just the first advice is quit it. But yeah, so you have to no, in the treatment, guys, obviously we have to administer oxygen, right, to displace all the carbon monoxide from the hemoglobin. That is something we have to do. Coming on to the next one, what is the porphyrias, right? So remember, the porphyrias are the metabolic disorders of heme synthesis. Now, remember, guys, there is increased excretion of porphyrins or porphyrin precursors. So there are two types of porphyrias, guys. We have erythropoietic and hepatic. So the enzyme deficiency, basically, it occurs in the erythrocytes, and the hepatic one, it occurs in the liver. Now, the porphyrias are basically, we have already discussed that, guys, right? So I will be coming on to first the enzyme defect, and then I will be discussing the heme wala part. Then it will be like more easy to know correlate with this. So we have the hepatic types of porphyrias and erythropoietic. Hepatic means the liver, guys, right? So we have acute intermittent porphyria, Porphyria cutanea tarda, Porphyria cutanea tarda, hereditary coproporphyria, and variegate porphyria, right? So the enzyme defect in acute intermittent porphyria, it is uroporphyrinogen 1 synthase, right? I will be coming to all these enzymes, right? I will be discussing the formation of the heme, and that is where you will know encounter if these enzymes are deficient, they will definitely result to these the disorders known as the porphyria, that is due to the decrease in the hemoglobin synthesis, right? The metabolic disorders of these heme synthesis. The porphyria cutanea, it is uroporphyrinogen decarboxylase, right? Okay, so the hereditary porphyritis, coproporphyrinogen oxidase, variegate porphyritis, porphyrinogen oxidase, right? So that is where like we'll be discussing it. Apart from that, the erythropoietic, we have the congenital. Congenital means beta by birth. By birth, we have the erythropoietic porphyria, and that is resulted due to a deficiency in the enzyme that is called as uroporphyrinogen 2 co-synthase, right? Apart from that, we have protoporphyria, guys. It is ferro um, Kilitis, right? Now, let's just try to discuss how the heme is synthesized. Then I'll come again back to this part, and we'll just now, guys. You already know the hemoglobin. It is basically made up of two components, right? The heme plus globin. Heme plus globin. Globin is just basically the protein part, and heme it basically comprised of ferrous and these pyrrole rings, right? The pyrrole rings, which are the porphyrin part, right? The porphyrin, right? Porphyrin and porphyrin 9 basically, right? Pyrinogen, right? So remember the most important porphyrin containing compound, it is the heme, right? So, okay, where this heme is synthesized, guys, in our body? Again, a very important question. It is synthesized in the liver. It is synthesized in the erythrocyte, producing cells of the bone marrow, right? The bone marrow produces erythrocytes, and that this is synthesized. Now we have different stages, guys. We have different stages. The first step, it is the synthesis of, yeah, the first step, or like let us just the first step, okay, the formation of S-aminolevulinate or I would say the first step, guys, it is always the synthesis of the porphobilinogen, right? Now, when I say porphobilinogen, guys, porphobilinogen basically it is a monopyrrole. It is a monopyrrole, guys, monopyrrole containing one of the single rings, right? Second is it is the synthesis or the formation of the porphyrin ring. It is the synthesis or the formation of the porphyrin ring. Apart from that, right, we have the chelation of this no iron, and right, we have this synthesis of heme at last from the protoporphyrin 9, right? So, hmm. Now, before coming to that is let us just quickly revise the heme synthesis, guys. That is the again the cycle that you have to remember. The succinyl coenzyme A, guys. The succinyl coenzyme A combines with glycine to form D-amino beta ketodipate, right, with the help of enzyme aminolevulinate synthase. Apart from that, the aminolevulinate synthase will again act to form the aminolevulinate, right? And guys, yes, again with the help of an enzyme aminolevulinate dehydrate, what will happen? Porphobilinogen will be formed. Porphobilinogen will be formed. And remember, guys, these porphobilinogen, right? These porphobilinogen, basically molecules. Now they will undergo conformational change by the help of enzyme uroporphyrinogen 1 synthase and uroporphyrinogen 3 co-synthase. And that is something that what they will form, that is uroporphyrinogen 3, right? Apart from that, they will form the coproporphyrinogen 3, protoporphyrin 9, protoporphyrin 9, and from protoporphyrin 9, what is formed? That is something called as the heme. That is something called as the heme. So you just have to remember in very short about the cycle. Mostly the disorders are important over here, right? Even they can ask you the cycle as well. So you just have to remember from succinyl coenzyme A, from glycine to remember all these enzymes. And again, if these enzymes are defective, that is something which will result in porphyrias, right? The metabolic disorders which will arise, no, which are helping in the synthesis of the heme. If these enzymes are absent or deficient, obviously these will result in the porphyrias, right? Now, coming on to the next one, that is the degradation of heme to bile pigments. Now, guys, you already know what is the lifespan of RBC. Lifespan of RBC, it is 120 days. After the lifespan is over, guys, erythrocytes are taken up and degraded by the macrophages, guys. They are degraded by the macrophages of reticuloendothelial system, that is the RE system. Now, hemoglobin is cleft. So what will happen? The heme will be degraded and the globin will be free, right? So the heme 80% it is the erythrocyte, okay, 20% it is the immature RBCs, myoglobins and cytochromes. So it will go into these sources, right? When we talk about the globin part or the globus part, it is again reutilized for the hemoglobin formation, or it can be degraded to individual amino acids. The protein, globin is a protein, guys. Protein is degraded to amino acids. So that is something you already know. Hemoglobin. Heme will be degraded to 80% of erythrocytes or 20% of immature RBC. It will no, it has two sources. It can either go to erythrocytes, or the 20% can go to know these immature RBCs, or it can be myoglobins, or it can be cytochromes. Whereas the globin part, it can be again reutilized for the heme formation, or this protein can be broken down into amino acid. That is simply the degradation to the individual amino acids of this globin part. Now, coming on to the degradation of the hemoglobin, guys, you already know if there is aged erythrocytes, if the lifespan of the RBC is over, right? Old RBCs, right? The aged RBCs, what will happen, guys? What will happen? This hemoglobin, no, this hemoglobin will be broken down by the help of no these heme and these protein molecules, right? Apart from that, these heme will be broken down by the help of heme oxygenase into biliverdin, right? This biliverdin, again with the help of an enzyme, biliverdin reductase will be converted into bilirubin. Will be converted into bilirubin. So this is how the series of no breakdown of this hemoglobin takes place, right? Heme plus globin will separate. Heme part, again with the help of an enzyme, heme oxygenase will convert into biliverdin. Biliverdin will convert into bilirubin by the help of enzyme biliverdin reductase, right? Now, what will happen, guys, next? Remember that this uh, in the blood, guys, in the blood, you already know this bilirubin will combine with an amino acid. Yeah. sorry, not an amino acid, yeah, the protein that is called as an albumin. So the bilirubin will be combining with albumin, which will result in the formation of this bilirubin albumin complex, right? And guys, after reaching into the liver from the blood, guys, they are going into the liver, and from the liver, what will happen? This bilirubin will be again free, right? From the no, albumin is something which helps in transportation of this bilirubin. Bilirubin alone cannot transport in the blood. It requires some precursors, like in the form of albumin, right? This protein is very, very important because it helps in the transport of albumin. No, it carries this albumin, means this bilirubin from the blood to the liver. Now, what will happen in the liver, guys? In the liver, with the help of enzyme bilirubin glucuronyl transferase, what it will getting converted into bilirubin diglucuronide, right? And that is something called as the bile, right? And this bile or this bilirubin diglucuronide, by the help of microbial enzymes, guys, that are actually present in the intestine, will be converted into urobilinogen, right? Now, in the intestine or kidney, it will be converted into urobilinogen. Obviously, in the kidney, it will be converted into urobilin, and that urobilin, it is basically excreted via urine. That is excreted via urine. Whereas if we come to the microbial enzymes, right, present in the intestine, guys, what it will lead to? It will lead to the formation of stercobilin, and that stercobilin will come out in feces, right? So that is something you have to remember. Coming on to the next part, that is the bilirubin hemosiderin, right? So just remember, guys, the basic chemistry about it. So basically, the breakdown of the normal heme catabolism, right? Now, you already know the principal pigment is in bile. That's why your feces are yellow in color, right? The yellow color. Okay. So that is that you have to remember. Apart from that, the transport part, it is in plasma inbound. Okay. And I've already told you it is bounding with which protein, guys? It is the albumin. Albumin is a very important protein, which helps in the bound of it helps in binding of this particular bilirubin, right? And that is something it is carried out through the liver from the blood, right? So this albumin bilirubin complex will help in the transport of bilirubin from macrophage to liver, right? So conjugation, basically in liver, you already know we have the enzyme bilirubin glucuronide transferase, which results in the formation of bilirubin diglucuronide. Okay, and you already know it is an active process. It's an energy dependent process where ATP will be utilized, and the fate, what is the result, guys? It will be converted into uropilinogen, which is like colorless and like is excreted urobilin, that is the yellow component, right? It's excreted and bile on the liver part, right? Remember, guys, the normal bilirubin levels, right? Whenever we do a liver function test, always there is something you will know, see in the blood reports, the normal levels of the bilirubin in the blood, basically it is 0.2 to 1 milligram per deciliter. And apart from that, the conjugated bilirubin, it is 0.2 to 0.6, and unconjugated bilirubin, it is 0.2 to 0.4 milligram per deciliter. Right, coming on to the next one, that is the jaundice or the hyperbilirubinemia. Now you already know hyperbilirubinemia. It is hyper means increased bilirubin in the blood, right? The increased concentration of bilirubin. Emia means the blood. Hyper means increased bilirubin, right? Symptoms is yellowish discoloration of eyes. Now, whenever a patient of jaundice will come to me, the very first thing which I encounter in the patient, the first clinical sign that I'll see over here, the sclera of the patient eyes will be no yellow in color, and that yellow color is basically coming from with this bilirubin, right? This bilirubin, right? So we have actually three types of jaundice that you have to remember, guys. The very first, we have the prehepatic, hepatic, and the post-hepatic jaundice. Over here, they have classified it as hemolytic jaundice, the hepatic jaundice, and the obstructive jaundice. The hemolytic, hepatic, and the obstructive jaundice. Now, remember, guys, in the hemolytic jaundice, it is something which is not happening in the liver. It is just before the liver part in the blood, guys. What is happening? The bilirubin over here will be unconjugated. Hepatic part, both conjugated and unconjugated, and in obstructive, if there is a obstruction in the bile duct or something, that will lead to the obstructive jaundice, guys. And that over here, that the bilirubin obviously will be conjugated. Now, over here, just you have to remember the bile salts. The bile salts will be present in the no the hepatic part, the obstructive part. The color will be echoluric in case of hemolytic, dark in case of hepatic, and like the same in kind of no this obstructive jaundice as well. Apart from that, what will be the stool color, guys? What will be the stool color, right? The feces will be in hemolytic, right? The dark brownish pigment, right? So the dark brownish pigment in cases of hemolytic jaundice. Okay, in cases of liver jaundice, the hepatic jaundice, the stool color it is normal, or it will be slightly lighter, the pale color, or in obstructive jaundice, it is clay color stool, right? The AST, ALT levels in the hemolytic ones, they are normal. ALP levels are also normal. Obviously, there's no defect in the liver, guys. It is just which is pre-hepatic, not in the liver. It's happening just before the liver. That is happening in the blood, guys. So the ALT/AST levels will be automatically normal. The alkaline phosphatase levels will be automatically normal. But if we come to the liver part, you guys already know this ALT enzyme will be increased and ALP will be increased 2 to 3 times. That is again a significant marker, guys. Whenever a patient of hepatic jaundice comes to me, if I do the LFT test, obviously the ALP levels will be rise twice or thrice automatically. Whereas when we talk about the obstructive jaundice, guys, remember the ALT/AST can be normal or it can be slightly variated, right? Apart from that, if we talk about the alkaline phosphatase levels, because it's no post-hepatic obstructively, it's there. You should know that it is 10 to 12 times, right? 10 to 12 times. Apart from that, urobilinogen, it will be present in the hemolytic and the hepatic component, but it will never be present in the obstructive component. And one more thing that you have to remember, the urine conjugated bilirubin, it will be absent in the hemolytic component. It will be present in the hepatic and the obstructive component. No. Coming on to the next one, that is jaundice due to genetic defects. So remember, we have the neonatal jaundice. That is something called as the physiological jaundice, guys. Now imagine mother coming with her baby in the first after the delivery. She comes within 2 to 3 days and she says, "No, my child is this and there is yellowish discoloration." So what is the first step? No, whenever a mother comes to me with newborn child or neonatal child, it is the normal jaundice, guys. You have to do nothing in that case. You have to just assure the mother, right? So that is something you have to remember in the neonatal or the physiological jaundice, guys. There is not true genetic defect, right? So there is defect in the UDP glucuronide transferase. It is initially low in the newborn, right? So what it will do, it will increase in the hemolysis of the RBC. Obviously, hepatic symptoms will be there. Okay, unconjugated bilirubin will be more than 25 milligram per deciliter. It crosses the blood-brain barrier and it can cause connectors. Now, in that patients, we actually start off with the phototherapy, right? We start with the photo. Initially, first 24 hours, way normal, guys, but after that is if it is continued for a longer period of time, then we have to do the phototherapy, guys. The bilirubin can absorb light up to 420 to 470 nanometers, right? So that is you have to remember this bilirubin. We actually keep those neonatal childs under the phototherapy under the light, guys. Now what this light do, this converts the bilirubin into lumirubin, and that is something which is excreted by the kidneys. That is something which is excreted by the kidneys, right? So there are various diseases, guys, that you have to remember. The very first one that you will study in medicine, that is the Crigler-Najjar syndrome. Apart from that, we have Gilbert syndrome, we have Dubin-Johnson syndrome, and Rotor syndrome. So these are certain disorders that you will be covering up in medicine part. But remember, these are all something which will know. Okay, in Crigler-Najjar syndrome, there is a defect in the enzyme that is UDP glucuronide transferase. Obviously, the bilirubin will be unconjugated, and there will be profound jaundice over here. In Gilbert syndrome, there is less activity of glucuronide transferase, unconjugated bilirubin, and the jaundice over here will be mild, guys. The Dubin-Johnson syndrome, there will be abnormal transport of conjugated bilirubin will be there, right? And the moderate jaundice is there. Even in the Rotor syndrome, the exact case the exact cause is not known up till yet, but it is believed that no, the bilirubin over here will be conjugated, and it is exactly the moderate jaundice which we can see. So these disorders, you can just remember the enzyme deficient over here. And guys, this is something they will not ask much about these disorders, but at least the enzyme defective, you should know. They can ask in practical as well. So that's all for today's session, right? So yes. So thank you, and we'll continue the session tomorrow as well. Thank you.